Hi everybody, and welcome back to the second session for today's workshop. This is where we're going to be hearing from Associates of Cape Cod. I'm delighted to be able to hand over to Veronica Wills, who's the Head of Technical Support with ACCI. She's going to be delivering a presentation. We're going to have a video to share with you, and then we're going to bring in her colleague Matt Stevenson to have the Q&A session. So please, as with the first session, please submit your questions and we will be taking those after the presentation and video. So without further ado, I'm delighted to be able to hand over to Veronica. Thank you.
Thank you very much, Jenny. Good morning, UK. Greetings from Falmouth, Massachusetts. I'm very excited to be here today because I get to share with you the newest developments that are happening in bacterial endotoxin testing. So we have a jam-packed agenda for this workshop, but before we get into the weeds of it, I just have to disclaim that any information provided in this workshop is for educational purposes only, and any decisions and actions, as usual, should be based on relevant regulations, guidance documents, and pharmacopoeia chapters. So today, I'd like to talk to you quite a bit about the bacterial endotoxin test. Some of you may be familiar with it already, and we're gonna go into details on how it works, what to expect from it, and then we were gonna be talking about the new sustainable addition to the plethora of bacterial endotoxin testing reagents. We're going to talk about how to validate those reagents and then how it all fits together. And then with the nice demonstration video towards the end, you're going to get a good feel for how it all comes together and how it works. So what exactly is the BET test, or as some people still call it, the LAL test? So that acronym stands for Bacterial Endotoxin Test. And it's actually a very interesting reaction, not a rocket science for sure, not as complicated and as involved as, for example, sterility testing, but it's much shorter, and it is actually considered to be a rapid test. Obviously, it's a test for endotoxin, and it utilizes a complex series of enzymatic reactions. And we call that reaction, or that complex, an LAL cascade. And to be honest with you, the entire test is about the cascade. The cascade is actually utilizing a natural blood clotting mechanism of this wonderful animal here that we're looking at, which is a North Atlantic horseshoe crab, Limulus polyphemus, and we're basically harnessing the blood clotting mechanism of this animal. It so happens that actually just down the road from me here in Woods Hole in Massachusetts, this reaction was discovered back in the 50s. And so at the time, what was discovered was that in the presence of endotoxin, the blood of the horseshoe crab starts clotting. It basically forms a jello. Today, we know in more detail that it's the factor C, which you can see in the cascade on the right side, that in the presence of endotoxin becomes an activated molecule, activated factor C, which in turn activates factor B, and that in turn activates the clotting enzyme. The clotting enzyme then reacts with a substrate, whichever substrate we end up using, which depends on the modification of the test. So this sequence of the three reactions leads to an amazing amplification of the signal, so amplification of the endotoxin present, so that for this fairly simple biological test, we're able to get to a very high sensitivity, down to 0.001 endotoxin units per milliliter, which is really picogram levels of E. coli endotoxin. So on this next slide, I'm gonna break down the substrates that are used for the various modifications of the test. So there's basically three different types of the BET assays. The original, and to this day still very often used, and also to this day the referee test here, is called the gel clot test. And you can see that in the picture on the right hand side, where the two hands are holding the reaction tubes. So what happens here is that this is obviously a manual, it's a visual test. We're going to be looking at whether we have a firm gel forming at the end of standard reaction time, standard reaction conditions, which is also temperature and pH. So what actually forms that firm gel is a coagulogen, which is the substrate. It's a very large protein, and that, after being cleaved by the clotting enzyme, becomes a coagulin. And the molecules, their increasing amount of the molecules in the reaction mixture, they actually start sort of coalescing, so kind of like getting stuck together. And eventually that forms the gel clot. So the hand that's holding, actually the right hand holding the tube upside down, that shows us what a positive reaction looks like. So that's a firm gel. The left hand is showing us the negative reaction, where at the end of the test, we don't have any reactivity. So you can use this test as a qualitative only, but you can also use it as a quantitative test if you test a series of dilutions, and then you can really come up with how much endotoxin is present in the sample. The next variation of the test is called the turbidimetric assay, and that's where we're also still going to rely on the same substrate, the natural coagulogen. But instead of waiting until the gel is formed, we're going to be measuring the output as a turbidity. So basically, we're measuring everything that happens in the reaction mixture before it becomes the gel. And that would be done with the use of an instrumentation and software. And on the right side, you can see an example of what actually those proteins look like in their freeze-dried state inside this is a standard 10 milliliter vial. So down we have the last version of the test, which is the chromogenic one. And here we have a difference. We're actually replacing the coagulogen with a different substrate. And this substrate is, we call it just generic as a chromogenic substrate, but it's a short peptide which has a chromophore attached to it. And the bond between the chromophore and the substrate itself basically gets cleaved by the clotting enzyme. So that's the last enzyme in that cascade. And that releases the chromophore. To the naked eye, that's going to look like a changing color, going from clear to this yellow color that you can see actually on the plate, because that picture is shown at the end of the test. And so that's basically what we're measuring. We're measuring the increase of the yellow color, so changing absorbance at 4 or 5 nanometers over time. And that would be done in the same instrumentation with the same software as the turbidimetric assay. So different types of products that we test, different types of, say, even medical devices would be tested with different types of reagents, and it all depends on how suitable the reagent is for the specific product. But let's first also talk about what is actually even endotoxin. So it's an exogenous pyrogen. So as any other pyrogens, and now our least favorite viruses, it will actually also cause a rise in body temperature. That's actually the very first symptom, but it can lead to other more serious symptoms as well. Endotoxin is actually fairly toxic to human bodies, and it's basically, it's a prototype of pyrogens. So we have a tendency to react to it. The problem is that endotoxins are derived from gram-negative bacteria. And you may know that gram-negative bacteria are ubiquitous microorganisms. They are everywhere. They pretty much have a PhD in survival. So they can adapt to any minimal environments, minimal growing conditions, including water systems. And that's where we could see an ingress of endotoxin over time. the endotoxin would actually be left behind sort of as a footprint of the gram-negative bacteria. It is not a living thing, as you will see on the following slide. It's actually a component of the wall of the gram-negative bacteria. So on the left, we're looking at a diagram of E. coli. And I wish I made that picture slightly bigger because you would have been able to see sort of the fuzzy outline all along the rod of the E. coli here. The fuzzy stuff is actually the endotoxin already. It's the polysaccharide portion of the endotoxin, which is what makes up endotoxin biochemically. The second portion, a large portion of the endotoxin molecule, is the lipid portion. And it's actually a phospholipid. And that's important here because you probably know that phospholipids make up membranes. So the phospholipid part is actually embedded within the outer membrane of the cell wall of, let's say, E. coli in this case. And that means that it's going to be actually an essential component of the cell wall. So it's pretty safe to say that every gram-negative bacteria will actually have endotoxin within it. And on the right side here, you can see the entire structure. So at the bottom, the red portion is the hydrophobic, the lipid portion, because lipids are always hydrophobic. And a typical, again, E. coli will have up to 7 fatty acid chains. So that actually gives it very strong hydrophobic properties. But at the same time, the molecule will also be hydrophilic, primarily because of the polysaccharide chain that is sticking out outside of the molecule, outside of the cell, but also because we have the phosphate groups attached. Actually, they're attached to the disaccharide, which is part of the red portion, and those phosphate groups sort of anchor the endotoxin within the cell wall. So long story short, it's a complicated molecule, and we do have to keep that in mind when we are working with endotoxin, whether it's a standard or whether it's a sample, because it will have implications on how the endotoxin behaves in an aqueous environment. Specifically, the hydrophobic portion will not like water, right? It's not going to want to mix with water, so it will have a tendency to create the large aggregates, micellar structures that will actually hide the endotoxin. They will have a tendency to underreport or even in extreme cases mask the endotoxin. So this is something we have to be very well aware of when we're working with it at the bench. And it's the lipid portion of the endotoxin that's actually the toxic component. So endotoxin is our bad guy. LAL is the good guy. Now, why do we even have to test for the endotoxin? So here in the US, we really don't have a choice. We have to. It's a cGMP requirement, and it is specifically mentioned in the Code of Federal Regulations for drugs as well as for medical devices. And I believe that it's pretty much the same situation over in the UK. What exactly do we have to test? Well, the requirement is for finished drug products and finished medical devices. We call them compendial articles. So any medical device that has a claim of being non-pyrogenic has to be tested for endotoxin. Any parenteral drug, so any injectable, has to be tested for endotoxin at release, every lot. But in addition to that, because of risk assessment, because of process controls, everyone is pretty much also testing any in-process samples, so in-process water, excipients, raw materials, so anything that comes in contact or becomes part of the finished product, it's actually tested upfront. And that's a good thing, because by the time we get to testing the finished product, it really should be just an exercise that we have to do, rather than coming up or running into surprises, at which point we would have to pretty much ditch the batch. The thing with endotoxin is It's not possible to get rid of it once we have it in the product. Well, okay, we'll rephrase that. It may be possible, but it would be extremely expensive to do so. So if we have a failed batch, a finished batch, it's pretty much the end of it for the batch. So let's take a look at how the testing actually works. So we have to follow the pharmacopoeia chapters, and there are 3 of them, and they're all harmonized. Like Paul was saying earlier, it's actually the Japanese pharmacopoeia, which is really into harmonization, and they are actually running this harmonization program for endotoxin as well. So we have to follow 85, and in the UK, you guys follow 2614, but they're basically the same, just saying the same thing slightly differently. In a nutshell, if we were to describe how the method really works, is that, again, endotoxin is the analyte. It triggers or initiates the reaction. The LAL cascade will amplify the reaction to the point that we're able to measure the trivial amounts of endotoxin by measuring either turbidity or absorbance. And then with the help of software, we're going to be able to back calculate endotoxin concentrations in our unknown samples. So let's see what the output looks like. And this is where it's going to get a little techy. So this is a typical plot of the LAL reaction, and this is extremely important. So number one, we're looking at a kinetic reaction here, meaning that we were measuring the optical density at set intervals. In this case, it was probably 30 seconds. And the beauty of that, as opposed to doing an endpoint test, the beauty is that we can see everything that's happening, because let's say our plate or the reaction vessel is sitting in the instrument, and the instrument is incubating and it's measuring at the same time, which is a great benefit, so that you can actually get a full movie of what happened rather than a single snapshot that we would get with an endpoint test. So in this movie, what I see is that, and this was actually a standard with a concentration of 0.5 EU per milliliter, which is pretty typical. So what I see is that at the beginning, there was nothing much happening. So optical density was very low, around 0. And that lasted up until we see this inflection here into the exponential phase. The exponential phase is the most important part here, because obviously that's where the signal is being amplified thanks to the cascade. Eventually, the reaction will saturate, because we're going to run out of the substrate, whether it's the coagulagen or the chromogenic substrate. So at which point we're not going to get any more changes in the absorbance. So this shape, sometimes we call it S shape or cobra shape. Different people see different things in this, but this is the typical characteristic of the reaction, and any other shape that happens during the reaction would not be specific to endotoxin. It would be a non-specific response. So when we are analyzing results, we always look at the shape as well, especially in the validation stages, to make sure that we're actually measuring the real deal and not something non-specific. So now we're going to look at what happens at different reactions. So here you can see basically a standard curve spanning from 50 down to 0.05 EU per milliliter, typical tenfold simple curve. But you can start to appreciate the differences in the reactivity with the various concentrations. So the more concentration we have, the faster the response to the endotoxin is, of course, because we have more endotoxin in there. So that is the light blue color on the left. You can see that the lag phase is very short, exponential phase very steep, and we get the reaction back very, very quickly. I did not mention on the previous slide that because of the amount of the data that we are looking at, we have to actually reduce the data. And the typical way of doing so is through reduction to onset time. And the onset time would be a time at which the optical density reached a specific threshold. That would be a validated value. And so for a 50 EU per milliliter, my threshold is going to be In this case, we have a threshold of 50 milliabsorbance units. So the 50 EU per milliliter will reach the threshold in probably around 400 seconds, so very quickly. But the 005 concentration is going to reach the same threshold you can see here in about an hour. And that's a very typical runtime for 0.05. That's a very fast test. It can get faster, as you will see later on. But you can see here that the different concentrations do react differently. And this is a standard curve, but we're going to see the same thing for samples as well. Now, we basically have 3 dimensions here. We are looking at time, which is in this case the x-axis. We're looking at optical density, but also a concentration. All of these are the variables. So obviously working with 2D plots only, we actually have to reduce this data again, and that's what the software does for us in order to plot what actually the standard curve is in the end. So the standard curve is onset time versus concentration. Okay, so basically how long it's going to take for each concentration to react. And because of the exponential phase or exponential nature of the reaction, we actually have to do a log-log fit. So we need to use a logarithmic function in order to get in here a nice linear plot. So the 50 EU per milliliter in this case is going to be on the right side of the plot, so at the very end of the x-axis, and 005 would be the left side of the plot. And so this is now the range of my standard curve. And so that's the basic. Now, if I want to calculate endotoxin concentration in an unknown sample, I am going to measure it. I'm going to get an onset time as my output and then plot that against the standard curve to come up with a back calculated value of endotoxin concentration. So basically, I'm interpolating against the curve to come up with what my unknown concentration is. And that's how it works. So again, nothing really, no rocket science here. It's going to get a little bit more scientific later on. So again, so the BET test is a crucial test. It's very useful for screening of quality. So it actually became a very useful quality indicator because it can get you rapid results. It's a microbiological output, really. It's a biological test, but we're speaking about, do we have any bacterial response in the sample? Did we have any gram-negative bacteria in there, which really are the most commonly found or most commonly ingressing bacteria? And so as a quick test, it provides the end user with very quick results so that they can make a decision on whether to accept a quality of a certain raw material or a step in the production process. So very, very useful. Now, this has been obviously in use and has been, a crucial quality test for over, I believe, 45 years since it became basically a compendial test back in the 70s. And today I will introduce you to a sustainable edition of a bacterial endotoxin test. And so the sustainable edition here is bypassing the need for using the horseshoe crab as the natural resource for the LAL reagent, and instead replacing it with a recombinant reagent. This recombinant reagent is actually called the recombinant Cascade reagent, and the word cascade in there, again, very important. It uses the same cascade mechanism as I was just showing you, but this time the cascade is actually recombinantly prepared. So every enzyme within the cascade was cloned and was expressed and then purified and then formulated into the reagent. Now, this is actually not that new because this reagent, which was already commercially released back in 2015 in Japan, is called PyroSmart. And at the time, it was subjected to a number of collaborative studies that were actually organized by the Japanese National Institute of Health. So those are actually, you have the references here on the slide. And then later on in 2019, there was another actually a large-scale study that looked at the suitability of this recombinant cascade reagent for testing of over 100 different injectable drug products. And they found out that it was actually working very, very well, that the interference pattern that we a lot of the times see for especially more complex drug products, there was actually a lower interference pattern. So in fact, PyroSmart was in many cases even more suitable for testing those drug products than the Compendial LAL test. But it gets more exciting because this reagent was really only available it was not globally available, that's the thing. It was only available in Japan. And so because it was shown to be successful, we decided to perform a technical transfer because it was actually being made by our parent company, Seika Gakko. And so we decided to perform a technical transfer of the production of the reagent over here to the US. And that is now becoming a next generation of the reagent, which we call PyroSmart NextGen. And we announced that last year, and it's something that is actually going to be launched later on this year. So why is it important that it's actually a recombinant cascade reagent? Well, the importance here is that it's the cascade is the front and center of everything. So the cascade is still there, which you will see on the following slide. And that also means, from that what stems, is that it's the same stuff. We're measuring the same entity. We are getting the same output. And that means that we can actually do everything the same way. We use the same instrumentation, same software, some preparation steps, and even the data interpretation is the same. So we don't have to learn any new tricks. And yet we get to use a reagent that is sustainable, but it also has other benefits on top of that. It is actually even faster, and we just really get to use the same existing method. And for someone in the QC, that's something that is always highly appreciated. You know, keeping things the same is in the QC world very important, because we all know how the QC labs get busy, and invention is not that appreciated a lot of the times. So let's take a look at the cascades here side by side. So on the left side in the blue, you can see the cascade that I was showing you earlier, so the same thing. On the right side in green, you can see the recombinant cascade. So the main steps there, the factor C, factor B, and the proclotting enzyme, they're all there again. But this time in green, those proteins are recombinant proteins. Now, there is a difference. So these cascades are not exactly identical, of course, but the difference is that the recombinant reagent is using only the chromogenic substrate, so there is no version with the coagulogen. And for a very simple reason, coagulogen is, as I alluded to earlier, it's a large protein that means that it would also have to be cloned and it would have to be expressed. The longer the proteins are, the more difficult that is. And we really didn't see a big value in that, especially because of the fact that chromogenic assay is actually a very popular assay and provides, you know, very good and linear data. So we decided that the chromogenic substrate is actually sufficient. So the recombinant cascade is a recombinant chromogenic test. Also, another difference, if you are careful here to look at these pictures, is that we are also actually missing here this glucan pathway. I did not mention it earlier, but glucan is actually another analyte that has a tendency to co-react with endotoxin in the LAL, in the natural reaction. And that's because the horseshoe crabs, when they live in the water, they have to fight against the gram-negative bacteria, which obviously are highly abundant in seawater, but also there's a lot of algae in the water. And algae contain, like their cell wall is basically made up of 1,3-beta glucans. And so for the horseshoe crab, this, the presence of the FG, which is a factor G, is actually another survival tool, so that in the presence of glucan, the factor G becomes activated as well. It basically is very similar to the factor B, so it also reacts with the clotting enzyme and basically causes the same reaction. In practice, for us in the QC lab, that means that when we're measuring endotoxin, we can also get results of glucan at the same time, not knowing that. So basically, it causes enhancement or a false positive reaction. And we are able to block the glucan response with the use of buffers. They're called endotoxin-specific buffers. So there is a way to bypass that. But with the recombinant cascade, you don't have to bypass anything because you're not going to even measure it because the factor G is not in there. So the cascade, the recombinant cascade reaction is truly an endotoxin-specific reaction. So how does the recombinant chromogenic method work? Well, again, it works basically the same way. So again, we're going to have endotoxin initiate the reaction. That usually happens in this. You can see the microplate, so it's a plate format assay, but other reaction vessels can be also used. And then through the amplification, so using the cascade, we're going to measure absorbance at 4 or 5 nanometers, let's say on a plate reader, and then the software will calculate the endotoxin concentration for us. So same stuff, same step. You keep the method, but you are going to get the benefit of the test. The benefit of the test, number one, is huge here already. The recombinant reagent is actually even faster reagent than the LAL reagent. So earlier on, I was saying that the typical assay is going to take us probably somewhere around an hour on the LAL test to get the results back. But that could be somewhat variable depending on the lot because it is a natural product. But with the recombinant version, you're going to be able to cut the assay time down by half because it's a faster test. So here in this table, we're looking at the onset times for both. And you can see that the onset times for the PyroSmart NextGen are actually shorter. So that means that the assay is faster. And so you get your results faster. That's the easiest way to put it, which is great. Again, being in a QC lab, we all know that fast is good. The faster, the better. But there are also other benefits to this test. So again, it's a sustainable reagent, so it no longer requires for us to even see the horseshoe crabs. We can come to the beach and see them there, but they would not have to come to us to donate a little bit of their blood. So that is definitely appreciated. Now, the next thing is that as a recombinant process, we also have a better control over the process. And so for you as the end user, that means that the lot-to-lot consistency would be much greater. So we will actually have a decreased test variability. The compendial accepted variability of the LAL or BET test is twofold. So everyone probably heard about the twofold error, and it means that we can only accept the results that are within 50 to 200% of each other. But I can tell you that with the decrease in the variability, that gap is now shrinking. So basically, we would be able to even get it down to 75, maybe to 150%, which is definitely appreciated as well. And you can see the impact of this also in the picture here on the right side, where we have onset time shown for 5 different lots of PyroSmart NextGen. And so these are standard curves for those individual lots, and they're basically all overlapping each other. And in short, that means that if they're on top of each other, they're the same. So these 5 different production lots are performing the same way. And that's great. That means that we, there's literally no variability in there. Another great asset of the test is that, like I was saying earlier about the glucan, it's not going to react with glucan because there is no factor B. And this is documented in the plot down on this slide. So you're looking at, again, standard curves. These are performed with the reference standard endotoxin, the RSC, 3 different lots. And the curve that's plotted on the x-axis contains actually the RSC with added glucan to it. It's a spike. So each concentration was actually spiked with 200 picograms per milliliter of glucan. And the curves on the y-axis were not spiked. And again, you can see that the curves are overlapping each other. So that means that they are identical. So that's actually also good. So it's a completely specific endotoxin specific test. So, and this is all achieved still under exactly the same quality assurance. The recombinant reagents in general, right now, they're not subject to FDA licensing. And you probably know that all of the LAL reagents are subject to LAL licensing. So, and that goes back to the fact that they're biological reagents. So, any LAL reagent has to be made under cGMP conditions. On top of that, here at ACC, we're also ISO 13485 certified. So we have those requirements on top of that. And we opted for actually manufacturing the recombinant reagent under exactly the same conditions. And so it's falling under the same strict criteria as well, so that the quality is there, just like it would be there for the LAL reagent. So we have obviously, a lot of data showing that this is a very promising future of endotoxin testing. And thankfully, the users or those that would actually be interested in using this in the field, they have been actually calling for this type of reagent for a while. The question is, though, how do we get to do it? How do we get to use it in the lab? So right now, the compendial environment around the use of the LAL reagent is actually very strict and stern, in a way that, it's something that was put in place, again, over 40 years ago. And change is always difficult in the pharmacopoeia. So the current status of anything that is new is that it is considered to be an alternative test. Okay, so the only compendial tests are those that are mentioned in the harmonized USB chapters, and those chapters mention only LAL or TAL reagents actually as well. TAL means that it's from Tachypleus, which is the Japanese horseshoe crab. So the use of recombinant reagents is considered alternative, and that's okay. Alternative reagents still can be used as long as they're shown to be, in short, equivalent or superior to the compendial reagent. And here you have a list of the global pharmacopoeias and their stance on this. They're all in line except for the European pharmacopoeia, which actually, beginning this year, they released their one new chapter for the use of recombinant factor C reagent, which is different from recombinant Cascade reagent. It's a fluorescence-based method. So that one was actually, it became a compendial method. However, all the individual monographs and general monographs in the European Pharmacopoeia will continue to refer to the BET chapter only. So to 614, they will not refer to the newest chapter, not for a while. So that means that if we want to use the recombinant factor C reagent for testing per monograph, so testing of a drug product per monograph, it would still be considered alternative test for that. So As an alternative test, like I said, we can use it, but we do have to test it in our labs to make sure that we would achieve the same results as when we would be using the pharmacopoeial chapters. And so how do we have to test it then? How do we have to show that? Well, there's 3 different components to it. Component number 1 is showing the analytical suitability of the new method. And that means here either validation following USP 1225 or following the ICH Q2 requirements to show the accuracy, sensitivity, robustness, and all the other requirements as well. I will show you actually example of that later on. The next thing is that we have to show that the alternative method is suitable for testing the drug products or the medical devices. And that means that we basically have to run a test for interfering factors, which is really the same thing as we would do for any compendial method as well. So that is not a difference. But there is another difference here, and that's the equivalency testing. And that means that I would have to show that my results with the recombinant method are actually the same as the result with the LAL method. So I'll show you just a few examples of this because we obviously did a boatload of testing and method validation studies. And I will just summarize some of that really quick here. So for example, looking at the analytical suitability of this recombinant Cascade reagent based on testing 5 different lots over a month, 6 different analysts, 5 instruments, 3 different labs. So very large type of study. It shows basically in the criteria column, you can see here what the criteria were, what we were shooting for, and then what we got. So the accuracy, the precision, the linearity was there. In fact, it was extremely repeatable, predictable. So we were able to not only meet the criteria, but in many cases actually even exceed the criteria in a way that we were consistently getting even lower values. So that all looks great. In terms of specificity, limit of quantitation, and range, that was done on a smaller scale with 3 lots and with 3 different analysts and so forth. But again, everything was confirmed, and we determined that the range for the onset time assay, you can see that there's also another assay that we can run here. It's a rate assay, but I did not touch on how that would work in terms of the data reduction. So I'll just stick to the onset time assay. So the onset time has the same range exactly as we would normally perform for the chromogenic and the turbidimetric assays as well, from 50 down to 0.05. And again, all of the other requirements were met as well. So from the analytical standpoint, the reagent is performing extremely well. There were many other studies that we performed, but for the sake of time, I'm just going to show you the most interesting data. And so, of course, everyone is interested, how does it work if I'm looking side by side as what would I get with the LAL test versus what I'm going to get with the recombinant test? So here we're looking at a panel of purified endotoxins. So these are, we call them lipopolysaccharides. They could be used as standards or they could be used as samples as well. But for example, the E. coli O55B5 is a typical standard. So what you're looking at here is what endotoxin activity or potency was measured by the recombinant reagent versus 2 LAL methods, chromogenic and turbidimetric, that we spoke about earlier. So first of all, notice that this is actually quite interesting. We will never exactly get the same number even with the LAL methods, right? Those numbers are fairly close. They are definitely within the 50 to 200%, which is the twofold error of the test, but they're not to be expected to be exactly the same numbers. So the variability is definitely there. And so what we did was we performed calculated a mean of the LAL assays, and then to calculate the so-called relative recovery of the recombinant method versus the LAL method. You just divide those two numbers, multiplied times 100%. And that is expected to be within 50 to 200%. So you can see here that the recombinant reagent was performing actually very, very well compared to the LAL method, with the relative recovery being well within that range. So that's great. That means that it's measuring the endotoxin. We're not going to under-report the endotoxin. That's the number one worry about any alternative method is, am I going to get the same result? Am I going to underreport? So definitely no underreporting here. And I can document that even further on these deionized water samples. So we chose to use those because obviously the lipopolysaccharide chemically purified version of endotoxin does not exist in nature. We wanted to use samples that contain endotoxin that does exist in nature, the endotoxin that we would be more likely to actually measure in the pharmaceutical production. So then that's kind of hard to come by because thanks to the GMP, most samples today are going to contain endotoxin well below the endotoxin limit, and most of the time it's going to be undetectable. So we wanted to use samples that will have detectable amounts of endotoxin in them. So these water samples collected from different points of use, from different facilities, and you can see the varying degree of the endotoxin in them, were tested again side by side between the recombinant and the chromogenic and the turbidimetric method. And we calculated the same relative recovery. And the recovery here is actually even, I would say, better in the sense that not only it falls within the 50 to 100%, but it also has a closer range to the 100%, which is always appreciated. And the bottom line here is also we tested a culture of E. coli. Apologize for this, E. coli endotoxin, which was recovered within 135%. So what we have here is that the recombinant reagent is definitely an alternative to LAL reagents. It was shown by us to be analytically suitable for the testing, and also has great platform for equivalency testing as well. So we believe that it's a great candidate to evaluate, but of course it doesn't stop here. It actually starts here. It, as an alternative test, it would have to be evaluated by anyone who is intending to use it in their facilities upfront to see how it compares, how it works on their samples, how it compares to their in-house LAL method. So how does this all fit together between the compendial LAL reagents and between this new addition to it? So the big picture is that the recombinant reagents are an option for a sustainability. Some companies, including ACC, we are very vested into sustainability, and we have actually a large sustainability program, part of which is actually one very specific program that I'll mention to you. It's we actually make horseshoe crab babies. It is our horseshoe crab sustainability program, where we are able, through a aquaculture permit, we are able to produce, to basically perform an in vitro fertilization of horseshoe crabs, and we use gametes from bait crabs, and that allows us to then grow them and basically cultivate, for the lack of a better word, these baby horseshoe crabs up until the stage where they're juveniles. They're able to bury themselves into the sand, and at that point we release them into the natural environment. We were able, since the inception of this program in 2018, to release back into the environment over 1 million juvenile horseshoe crabs. And this program is now also expanded into Asia. And that's actually is very important because they really struggle there with the the Japanese horseshoe crabs and Singapore, the Carcinoscorpus, they are endangered species for sure. And so this program is now started here as well, so that they can actually bring, they can give back. We love to give back as well. There is more information about this on our website, beautiful pictures, which I always like to look at. So you can actually see these juvenile crabs here on these wonderful pictures. So that's one way of us giving back. And so we obviously use the natural resource because the blood of the horseshoe crabs serves, and it's supposed to serve as the source of the enzymes. But with this new addition of this new reagent as a recombinant cascade reagent, there is a way to bypass that as well for those for which the sustainability is of importance. So before that can happen, though, before we can really fully transition over to an alternative test, there have to be really wide comparability study. And that's something that we have been working on for a while now. We are in the process of collecting samples from various sites, from various customers. We also have an evaluation program that you can join if you're interested in seeing how this new sustainable reagent works. But ultimately, the point is that we're going to be able to show how the recombinant reagent compares in a variety, a plethora of different samples to the LAL reagent. So with that said, you can find more information on our website. We have a dedicated website to PyroSmartNextGen.com. But with that said, I would actually love to now show you a video. It's a demonstration video where you will get to see, actually it's kind of 2 for 1. You will get to see how the LAL test is set up, and we're going to focus specifically on the use of the sustainable recombinant reagent within this. So we will get the demonstration video up and running.
Hi everyone. So now we have the opportunity to take some questions, and thank you very much to Veronica for her excellent presentation. And joining us now is Matt Stevenson from ACCI as well. So welcome, Matt. Veronica, did you have some commentary that you wanted just to add in relation to specific elements of the video before we get into the questions?
Yeah, just shortly. So in the video, we were looking at how to set up very quickly the Kinetic Chromogenic assay. And that is the same format as we would be using for the LAL reagent as well as for the recombinant reagent. So you kind of got the 2-for-1 because you saw the LAL test, but with the use of the recombinant Cascade reagent in it. So thank you.
Perfect, thank you. So we have around 10 minutes or so for some questions. So I'm just going to take the first one that's come in. If there are any other questions that our audience have, please feel free to submit them and we'll pose them to our speakers. So first question is, with the development of recombinant agent, can you see a time where we may be able to test endotoxin in line for WFI systems?
That could be definitely development for the future. So for the inline testing, you would have to obviously, do that preferably on the floor. So with the use of various, specifically developed devices for that. Right now, the idea is that the reagent can actually be used on any system that will measure a chromogenic assay. And there are actually new systems out there right now that kind of already are thinking down the line of the inline testing as well. So definitely subject to further development and further advances.
Just to add to that, Veronica, as well, I mean, it does depend on the capacity as well of testing. I think Veronica's covered it really well in terms of the test is rapid anyway. Sensitivity down to 0.001 within 1 hour. Obviously, with PyroSmart and NextGen, we're looking at reducing that down further to around about 30 minutes. So it's kind of not one size fits all really in terms of water testing. It tends to be a larger capacity. So those types of traditional systems that you have in your laboratories already, the absorbent systems where you would see a greater throughput and within a shorter time space as well.
Okay, thank you very much. Just moving on to the next question. What is the British pharmacopoeia on recombinant agents for bacterial endotoxin testing?
I believe that they are in line with the European pharmacopoeia. So the notion would be the same, that the recombinant reagents for now are alternative reagents. And with the appropriate validation systems, it would be brought online then. So. But this is definitely something that's a very dynamic situation, changing almost monthly, I would say, and we're staying on top of that, and we'll be also continuously updating the audience and the end users around these changes. So we often run webinars with further information on this as the landscape is evolving.
Mm-hmm. Okay. Thank you. We do have another question. What is the difference between recombinant factor C and recombinant factor Cascade reagent?
Good question. I can take that one, Veronica. So in terms of formulation, if I start with recombinant factor C, I think Veronica has gone through in detail the natural LAL cascade and the different factors that are involved. A recombinant factor C reagent, if we look at that formulation, it's just really one of those factors. It's factor C. Factor C is a bio sensor that detects. It's the first marker within the cascade, and that will bind to endotoxin. But that is a single step process. You don't get the same amplification as what you would get in a LEL reagent. what you would see in the case of PyroSmart NextGen, fully recombinant LEL reagents. So you have to amplify the output in some way. And that typically involves a separate piece of equipment where you would need to amplify and enhance the reads that for recombinant factor C using a fluorescence reader. There are some differences as well in terms of the method preparation between the two reagents. One is a kinetic reagent, which is PyroSmart NextGen, which is the same as what most laboratories will be using. Recombinant factor C is an endpoint test. So in terms of the preparation steps, there are some differences there between the two methods as well. PyroSmart NextGen, I'm probably covering old ground here in terms of Veronica's already covered it, but you could see that as a fully recombinant LEL reagent. We're calling it recombinant Cascade reagent, but basically it mimics the natural Cascade. So like Veronica alluded to in the presentation, that means that you have all the same preparation steps that you have currently with your endotoxin testing in your lab.
Okay, thank you very much. We have another question coming in. You highlighted great benefits of the recombinant reagents, but how does the cost compare to the native reagent? Could this be a barrier?
I can pick that one up, Veronica. Cost is comparable to the current chromogenic reagents that are in use at the moment. So in terms of our chromogenic reagents, we sort of modelled it to be equivalent to those prices. So there shouldn't be any cost differential in terms of an increase when it comes to our reagents, at least when you're looking at recombinant technology.
Okay, thank you very much. Let's just have a look. From a viewpoint of, let's just see, one other question coming in. I think you would have covered this to a certain extent, how are the recombinant agents produced? Just to provide some clarification.
Okay, I'll take that. So yeah, they're recombinantly produced. So basically we cloned the DNA sequence of the North Atlantic horseshoe crab, so that's Polymylus polyphemus. We cloned that and created 3 individual cDNA clones, so one for factor C, one for factor B, and one for the procoding enzyme. These clones are then transferred into pretty much vectors and into an expression system, which is going to express the proteins responsible for the cascade. So then the proteins go through a purification process and then are formulated into the final reagent. So obviously that process, the manufacturing process is different, but this is the same process that would actually be used for any other type of recombinant, even drugs. For example, insulin these days is produced recombinantly only and follows the same idea, right? It's not the same process, obviously, but it's the same workflow, essentially. So in that sense, it's a standard process. It's the standard technology used these days from the master cells. And what it gives us is actually more control over the proteins that are actually present in the final formulation of the recombinant reagent. So hopefully that covers it a little bit.
Okay, well, thank you very much. Veronica and Matt, is there anything else you would like to comment on? Because that kind of concludes it, the questions that have been submitted so far. So anything else you would like to have the opportunity to share?
Just from me, Jenny, just a quick thank you to the PHSS for inviting us to speak. A special thank you to my colleague Veronica for getting up extra early on the east coast of the US. And to all the delegates as well. I just, if anyone does have any questions, we've put some information on our exhibition area, and there's contact links for each of our regional technical support experts. So if anyone's interested, has any questions about what we've covered here, would like to discuss anything further, then you can click on those links for your local expert. You can book in some time in their calendar, and that's available in the exhibition area on the platform.
Brilliant. Thank you very much. Well, thank you very much both for your time today, and also to our speakers from earlier this morning, so Paul Beckett and Adina Pound. Again, once again, apologies for some of the technical issues, but I think we've still been able to bring you those practical insights and that fantastic level of information today, and also throughout the rest of our aseptic processing and contamination control workshop. I would say a huge thank you to all of our subject matter experts, our sponsors, and everybody behind the scenes who has been able to bring this together. We hope that you have found these sessions informative, educational. You can come back and watch them at a future date. So if there are things that you wanted to kind of refresh, you can do that. If there are any questions that you have either for the PHSS or any of our subject matter experts, then please feel free to get in touch with the PHSS, and we can direct those to where they need to go. But once again, thank you to everybody who's been involved in preparing these sessions and facilitating them. Thank you for your attendance and engagement. Thank you to everybody who supported us and continues to support the PHSS. We endeavour to ensure that we can bring you the most relevant and kind of up-to-date information on a whole variety of topics. So thank you once again. Thank you to everybody. If you can, please give us your feedback. We will be sending a short survey out at the end of today's session. We do value any feedback and suggestions you have for this event, but also for future events as well. Obviously, we've been adapting to the virtual environment, and we will continue to do so to make sure that we can still provide the opportunities to connect with the subject matter experts and industry. So thank you very much, everybody, for your time and your engagement. Thank you for the questions. Thank you to our speakers once again for today, to Adina, Paul, Veronica and Matt. And please keep safe, keep well, and enjoy the rest of your day. That concludes the end of day 4 and our workshop series. Thank you.